Hello everyone, so my name is Sarah and today we are going to talk about vitamin C and how it can do different functions and what is the history about it and what are the things that are important to know. So first of all, we all know vitamin C is a um nutrient that is widely available in fruits and vegetables, and we need it because human cannot actually synthesize inside their body, unlike many other um, organisms. So first of all, this is because we lack an enzyme that is gluconolactone oxidase, which actually is important for its synthesis, and that enzyme is actually present in other organisms. That's why they can actually prepare that. Secondly, if we talk about its properties, it is oxidized by high pH, oxygen, or high temperature. So it is very sensitive in solution form. This is what I'm talking about in solution form. Uh, but if it is in powder form, it is more stable than solution. And there are two basic uh, forms inside the body in the biological system, that is DHA, that is dehydroascorbic acid, and the other one is ascorbic acid itself, or ascorbate. So both of these are present in the biological system and they are actually interchangeable, but they have similar function because they are interchangeable. They can convert into one another, but they require different transporter to get into the cells. So for example, ascorbic acid, it's going to need CBCT, which is sodium dependent transporter. So it will need this transporter to get into the cells and do its function over there. Whereas the other form, that is DHA, it requires another transporter, that is glucose transporter or GLUTs transporter. This actually takes in the DHA and and then it is converted into ascorbic acid through an enzyme present inside the cells, and then it can act interchangeably. So this is the introduction. And next, if we talk about the physiological concentration present inside the human body. So in women, it is around 75 milligram, whereas for men, the recommended dose is around 90 milligram for daily dose. And this can easily be obtained from a healthy diet. Like, it is widely available in fruits, for example, your oranges, and all other citrus family fruits. So it is very easily available if you have healthy diet. And then if we talk about the physiological plasma concentration of vitamin C, it is around 60 to 100 micromolar per liter. We all are aware of its functions, such as neutralizing ROS. So ROS is reactive oxygen species, which are being produced through several uh, mechanisms inside our body, and it is totally normal um, for body to produce ROS. But if there is too much, that could be because of some damage or some kind of mutation or some kind of disease, then it could be problematic. But otherwise, we are producing ROS on a daily basis, and it has to be neutralized by agents like vitamin C itself. Other than that, it has other functions as well, such as maintenance of collagen and synthesis of muscle keratin, and several other functions like cofactor for dopamine and beta hydroxylase. So that's the enzyme, and it acts as a cofactor for that enzyme, and then cause the formation of norepinephrine. After that, if we talk about hormones, we also require vitamin C for them to show their maximum activity. So over here, I have this figure. So over here, you can see there is the production of ROS, and then this vitamin C is going to do its function of antioxidant, and it is going to neutralize this reactive oxygen species, and then it will itself get um, oxidized and reduce the other reactive oxygen species. And now you may think that even if it neutralized this one, now this is another reactive oxygen species. So how this does not cause any harm to the body? So the answer to that is like we have different mechanisms in the body for the regeneration of this species back into the ascorbic acid. And also because of the structure stability, it is very less harmful as compared to this one. Further, if we talk about the vitamin C transport, so all of this is just to give you introduction and the basic knowledge about vitamin C and how it functions normally, and then we will move toward the next part, which is the title of this video is about. So vitamin C transport. So first of all, we have vitamin C in our body. We take it, and it goes into our gut, right? So over there, it get absorbed into the cells of intestines or kidney to simplify it. And we have the transporter that is CVCD1. It's gonna go into with sodium. So this is co-transport, and then it is going to go into the blood. This is the absorption uh, part. So now it is in the blood, and it is going to be supplied to going to distribute into different parts of the body, for example, brain, or for example, other regions like lungs, eye, bones, ovaries. So once it's into the CSF, that is cerebrospinal fluid, now it can go into brain, uh, and it will go to neurons and astrocytes. And neuron actually take a very high amount of vitamin C because they are 24/7. They are functioning 24/7, and they are highly active cells. So they are producing a lot of reactive oxygen species on a daily basis. So they need more antioxidants such as vitamin C to normalize or to neutralize those reactive oxygen species and to keep on working. So that's why you will see here that there is such a huge amount of vitamin C present in a neuron, that is about 10 millimolar. And because astrocytes also help neurons to maintain or to supply nutrients, so they are actually also having some amount of vitamin C, and they also actually help neuron to recycle the ascorbic acid that they have, and they have already utilized, and now they want to convert that back into ascorbic acid. So astrocyte do that for um, neurons. Like over here you can see DHA is produced, so it is excreting that, and astrocyte is taking up that DHA and converting that back into ascorbic acid and giving that back to the neurons. So it is helping neuron to do its function. That's why we call them helping cells. So this was the basic overview of vitamin C and how it does its transport throughout the body. So I have explained brain distribution more because it is a lot different than the other region because of the blood-brain barrier and everything else that brain has different than other parts of the body. That vitamin C could help uh, cancerous cells. Um, So these are two guys, Cameron and Pauling. So these two guys actually suggested the idea that vitamin C could kill cancer and could be helpful in killing cancer in when it is given in higher dose. And they conducted some trials, and they were very persistent about it. But when the other guys, other people's or other scientists tried to do the same, they were unable to replicate those results. And eventually, long story short, the idea that vitamin C could be used as a therapy for cancerous um, cells just faded. But I will cover this whole story and how they conducted their trials and how other people conducted their trials and what actually happened in those 10, 20 years. I will cover that in next video. But for this one, I'm gonna go just into the scientific part of it, not into the history that how they did it and what were their results. So eventually, of around 20 to 30 years, some other scientists, when they conducted clinical trials, they found out that vitamin C concentration was different when it was given through oral route versus when it was given through venous intravenous route. So here are the clinical study. Like over here, you can clearly see, like uh, if they were giving oral dose and they were increasing the dose. However, when they were increasing the dose, the concentration in the plasma remained constant. It did not exceed it over around 200. And versus when they give vitamin C through IV route and they increases the concentration, and they found out that vitamin C concentration in the plasma increased significantly when given through IV route upon increasing the uh, initial dose. For example, in 1 gram, first they give the 1 gram dose, and they ended up giving 100 gram, like 100 times more, and they found out like there was significant increase in the plasma concentration. Over here, you can see it's just like I cannot even see it. Like it's very basic, like less than 1 millimolar, right? And sorry, and upon giving like 100 grams, they could see around 15 millimolar concentration of vitamin C in the blood. So how that difference was occurred, and this difference is not only significantly big, but also it was shocking that how this much difference could be occurred uh, just by different route. So next on, they found out there were some uh, vitamin C transporter that I already told you about, that how vitamin C gets into the body and distribute towards different tissues or organs. So they found out that these vitamin C transporters are very selective, and they actually get saturated when you increase the concentration of vitamin C. First of all, if we talk about the factors that limit the absorption, there is intestinal absorption or bioavailability declines at doses greater than 100 milligram. And why is that? Because there is saturation and also downregulation of these receptors. Later on, if we talk about the renal absorption, reabsorption, so that is also saturated, uh, and that actually cause the excretion of ascorbate through urine. So there are multiple factors which could influence the absorption or retention of the vitamin C inside the blood. And when we give this vitamin C through IV route, all of these factors are eliminated. Now we have vitamin C directly into blood. We do not have to pass through any of the transporters. We do not have to deal with any renal reabsorption, saturation, and everything is cancelled out. So we have a lot more than that what we have through oral route. So later on, people actually saw that these are the things that are different in different routes. Now, they further investigated that if there is such a big difference in the concentration, there has to be difference in the role of vitamin C, like a dose dependent role of the vitamin C. And they were right, like just like Cameron on polling suggested, there was anti-cancerous effect. But how that anti-cancerous effect was actually happening in IV route. So vitamin C, when it is given in higher doses, like very high doses, reaching up to millimolar concentration in the plasma, that concentration actually cause the production of reactive oxygen species rather than so vitamin C actually act as pro-oxidant rather than antioxidant. But this action is very selective in the cancer cells due to different factors, which I will talk later on. First of all, vitamin C in the form of ascorbic acid, when it is delivered through the blood in the and it goes into extracellular spaces, and now we have this cell over here. So in the extracellular spaces, it is being converted into ascorbic radical, and the electron from ascorbic acid actually convert the uh, ferrous into ferric, and then it caused the formation of superoxide, which ultimately caused the formation of very uh, very harmful reactive oxygen species, that is hydrogen peroxide. And this hydrogen peroxide is very uh, permeable to the permeable uh, to the cell, and it can go into the cell and cause damage over there. It can cause DNA damage. It can um, cause mitochondrial DNA damage, and it can also cause the decrease in energy indirectly, and then ultimately everything in the cell will go down and causes cell death. So this has been already discussed in several studies by now, and uh, these are all the things that vitamin C in higher dose can cause. So all of this collectively together cause the pro-oxidant action of the vitamin C. However, this only occurs in cancer cell. So selectivity is due to different factors, and one of the factors that is widely been discussed in several research articles is that vitamin C, normal cell have catalase enzyme, which is one of the enzyme which cause the conversion of reactive oxygen species into harmless or harmless species such as water and oxygen in the case of hydrogen peroxide. Whereas in the cancerous cell, this catalase is not only its functionality not only reduced, but also its number is reduced. So both of the factors, like its functioning as well as its number, is reduced. So now there is more hydrogen peroxide, and it's gonna cause the death of the cell. So it is widely studied till now that the degree of down regulation and the reduced ability of the catalase um, is present in different cancers. However, the how much catalase is effective or functional in the cell is different between each kind of cancer cells. And the factor that may influence vitamin C uh, selectivity toward cancer cell is that the availability of the labile iron pool. So the general concept is that labile iron pool is more in cancer cells because of metabolic disturbances, because cancer cells are dividing rapidly. They do not have fully functional organelles, and they are having increased labile iron. So when there is increase in the production of hydrogen peroxide by this vitamin C, um, it actually goes into the cell and cause not only damage, but it also cause um, another production of another reactive oxygen species, that is hydroxyl free radical, which further enhance the oxidative damage. So now there are two culprits inside the cell for causing the oxidative damage. Not only hydrogen peroxide, but its but another of its partner, that is hydroxyl free radical. They both together cause significant damage, and then it can ultimately cause death of the cell by causing irreparable damage to lipids, DNA, protein. And how it is produced is because there is excess iron present in uh, cancerous cell. So iron actually, when combined with when cause the reaction with hydrogen peroxide, is gonna produce this species hydroxyl ion, and then it's gonna cause the damage. And this iron is actually present um, on these biomolecules. So it is site specific damage. For example, the iron is attached somewhere near the structure of lipid or DNA. And then when hydrogen peroxide is going to come over here, it's going to bind, it's going to attach, or you can say react with that iron. And then it will cause the production of hydrogen peroxide, sorry, hydroxyl ion over here. And because it is so near to the lipid, it's gonna cause the damage to the lipid. And this is site specific reaction or damage. And this is very dangerous uh, for the biomolecules, because it is not repairable, because cell is already having so much stress because of this species, that it is unable to um, clarify the damage that has been done by hydroxyline free radical. Sorry. So there are studies where they use the chelators, and it actually attached to the iron and then inhibit the formation of hydroxyl free radical. This way, we got to know that how much oxidative damage is caused by hydroxyl free radical, and how much damage is caused by this hydrogen peroxide. So we know how much contribution they are doing towards cell death. So this reaction is actually causes phantom reaction, and it is significantly important when we study vitamin C. So what's next? So vitamin C is currently under investigation in clinical phase 2 trial. So there are a number of studies ongoing, like on pancreatic cancer, on glioblastoma, on lung cancer, and several other. So scientists are doing several investigations on vitamin C effectiveness on these cancers, and if we get to know that it actually works well or significantly well, we can actually combine this with other chemotherapeutic agents, and we can get to know that we can get to have better effects. Because vitamin C is not only cost effective, but it is also widely available around us. It is everywhere, but we need to just use the high amount and through intravenous route, obviously, because otherwise it will not be effective. It will not reach to the that high concentration which is actually required for its pro-oxidant effect. So that's all about it. And another thing that vitamin C, we also think that vitamin C could have like lesser side effects as compared to traditional chemotherapeutic agents because it's naturally produced, and also because our body also utilize it, um, but in lower dose, obviously. Thirdly, because it is selective towards cancerous cells. That's another thing that could be very beneficial for cancerous patient. So that's all about it. In the next part, I will go into the details of these guys and how they found out that vitamin C could have anti-cancerous activity. They did not know anything about pro-oxidant, but they had the opinion through their trials that it has some anti-cancerous effect. And eventually, the other people who did some trials, we will get into all of that in the second video. Till then, goodbye.